Imagine you're standing in the produce aisle, staring at a pile of ripe, vibrant tomatoes. You'd swear that redness is as real as the skin itself, a physical property of the fruit, sitting there under the bright fluorescent lights. You might even feel like your eyes are reaching out to grab that color, scanning the shelf like a searchlight. It's a common intuition. In fact, research shows that nearly half of all adults believe our eyes emit some kind of energy to help us see the world around us. We feel like active participants in the light, projecting our vision onto the objects we encounter. But then you look at the person next to you, also reaching for a tomato. You might have that classic late-night philosophical thought, is the red I see the same as the red they see? You ask them, and they nod, confirming that tomato is indeed red. You leave the store satisfied that you both live in the same colorful reality. The truth, however, is far more unsettling, that tomato isn't actually red. In the physical world, there is no such thing as red or blue or yellow. There's only a chaotic soup of electromagnetic waves of varying lengths bouncing off surfaces. The vibrant scarlet you're so sure of doesn't exist on the tomato, nor does it exist in the light hitting your eye. It is a fabrication, a biological interpretation that exists only within the dark, silent theater of your brain. We are essentially translating a tiny, invisible sliver of energy into a personal light show. And while we've spent centuries debating whether we're all hallucinating the same rainbow, the way our brains actually construct these colors is a feat of biological engineering that scientists managed to map out 100 years before they even had the technology to see the cells responsible for it. To understand color, you have to stop looking at the world and start looking at the way your mind invents it.
It's a pretty heavy way to start, isn't it? That everything you see is a lie.
Well, not a lie, but a fabrication, an invention, and makes you question everything.
It should. We walk around completely convinced that color is an objective fact.
Yeah.
You know, like the weight of a stone or the height of a tree.
Right.
But the moment you start peeling back the layers of physics and biology, that certainty just it evaporates.
It's gone.
Color isn't what's out there. It's the code our brain wrote to help us survive what's out there.
Okay, I want to stick with that code idea because you mentioned something in the intro that, I've got to be honest, it sounds fake. It says nearly half of adults believe eyes emit energy. Like, we think we have Superman heat vision. That feels like a statistic that just can't be true in the modern world.
It sounds wild, but it's a very robust finding. The study mentioned by Wiener from 2000 showed that even among educated adults, there's this implicit belief in extramission.
Extramission.
It's the feeling that vision is an active process, like a searchlight sweeping a room. And if you think about it experientially, it makes sense.
How so?
When you look at that tomato, it feels like you are touching it with your gaze. You feel like the agent in the process.
I guess I do. It's hard to accept that I'm just a passive bucket collecting photon.
But that's physically what's happening. Vision is reception. You are being bombarded by electromagnetic waves and your eyes are just catcher's mitts.
Okay, so the tomato itself, it's not red. What is it doing?
It's absorbing short and medium wavelengths of light, and it's reflecting the long ones. We're talking roughly 600 to 700 nanometers.
So those specific waves hit my retina.
And only then, only inside your head does the red happen. If you weren't there to look at it, the tomato wouldn't be red. It would just be a lump of matter reflecting 700 nanometer energy.
This leads to the real headache-inducing part, the inverted spectrum problem. I remember arguing about this in college dorms at 2 in the morning.
Oh, it's a classic. John Locke's famous thought experiment.
Right. If the red is only happening in my head, how do I possibly know it looks like the red in your head?
You don't, and you never can.
But wait, if I show you a fire truck, we both say it's red.
Of course. We can verify that we both discriminate the colors the same way. If I show you a color chart, we both point to the same ship and say red. Functionally, we're aligned.
Okay.
But the subjective experience, the qualia, the actual feeling of redness, that remains a private screening. My red could look exactly like your green, and as long as we both call it red consistently,
we'd never, ever know.
Never.
That is a terrifyingly lonely thought. But since we probably can't crack the hard problem of consciousness today,
probably not.
Let's look at what we can crack. The machinery. I want to get into the dark theater you mentioned. I always thought eyes were simple cameras. You know, lens, film, picture. But the notes suggest the wiring is, well, bizarre.
Bizarre is an understatement. The retina is a masterpiece of counterintuitive engineering.
Okay, break it down for me.
So you have the rods, about 120 million of them, which are your night vision, your motion detectors. They are colorblind and blurry, but incredibly sensitive to light.
The black and white system.
Pretty much. Then you have the cones, only about 6 million of them, clustered right in the center in the fovea. These give you your high definition and, of course, your color.
So far, so good. That sounds like a camera with 2 different film types.
It does. But the weirdest part isn't the cells themselves, it's how they talk to the brain.
This is the part about them being on in the dark.
Right.
I had to read this 3 times to believe it.
It goes against every assumption we have about how sensors work. Usually a sensor is off until it detects something, then it turns on.
Right. My doorbell camera doesn't record until someone walks by.
Your eyes are the opposite. In total darkness, your photoreceptors are depolarized. They are burning energy, actively pumping out a neurotransmitter called glutamate.
So they're shouting into the void.
They're screaming at the brain, nothing is happening, it's dark.
Wait, so my eyes are working harder when I'm asleep with the lights off than when I'm looking at a bright screen?
Metabolically, yes. This dark current, as it's called, is incredibly expensive. The retina consumes more oxygen by weight than almost any other tissue in the body, largely because it has to maintain this constant electrical shouting when there's no light.
Why on earth would evolution design a system that wastes so much energy when it's not being used? That seems like terrible design.
It seems inefficient on the surface, but it's actually about speed and sensitivity. Think about it. If the cell is already on and sort of vibrating with tension, it can react to a single photon instantaneously.
Okay.
When light hits the cell, it hyperpolarizes. It shuts the gate.
Yeah.
The flow of glutamate stops. The brain detects the drop in the signal.
So the signal isn't the light, the signal is the change.
The signal is the silence. It's like holding a car horn down continuously, and the only way to signal I see something is to let go of the horn. The sudden quiet is the message.
That is intense. So we are detecting the interruption of darkness. Okay, so this signal, or this lack of a signal, travels from the eye to the brain. We have 3 types of cones, right? S, M, and L.
Short, medium, and long wavelength sensitive cones. We colloquially call them blue, green, and red, but that's actually sloppy shorthand.
Why is it sloppy?
Because, for instance, the red cone, the L cone, doesn't actually peak in the red part of the spectrum. Its peak sensitivity is in the yellowish green.
Hang on. If the red cone peaks in yellow-green, why in the world do we call it the red cone?
Because it's the only one of the 3 that is still sensitive way out in the long red tail of the spectrum.
Yeah.
But this shows you the eye isn't sending a red signal to the brain.
What is it sending?
It's sending a data point, a ratio. It's saying L cone is firing at 80%, M cone at 50%, S cone at 0%. The brain receives these 3 numbers and has to calculate what color that implies.
It's basically doing algebra constantly.
Nonstop algebra. And the signal doesn't just land in one place. It travels to the back of the brain, to the visual cortex. Specifically, an area called V1 first, then V2, then V4.
And what happens in V1? Does it see the color?
No, V1 is strange. It acts like a sketch artist who only cares about edges, orientation, and motion. It doesn't really care about color, it just outlines the whole scene.
So the first stage of vision is a black and white sketch.
Essentially, yes. Then that information is segregated and sent to other areas, and the color information specifically gets processed in V4, which is often called the color center.
The color center, okay.
And this is where we find these very particular structures that researchers somewhat unpoetically named globs.
The globs of V4 sounds like a terrible sci-fi movie from the 50s.
It really does, but these millimeter-sized clusters of neurons are crucial. They don't just register the wavelength, they create something called color constancy.
Color constancy.
Think about it. A red apple looks red in bright white sunlight, and it still looks red in dim yellowish candlelight.
Right.
Physically, the wavelengths bouncing off that apple are completely different in those 2 situations. But your V4 compares the apple to the background and says, relax, it's still the same apple, keep painting it red.
So the brain is color correcting the video feed in real time.
Constantly. Without V4 and the globs, the colors of the world would shift wildly every time a cloud passed over the sun. You'd never recognize anything.
That makes sense. It stabilizes our reality. But there's another limit to our processing, the fact that we can't see certain color combinations. The opponent process theory.
Right. So while the retina works with 3 colors, that's trichromacy, the brain organizes them into antagonistic pairs.
What are the pairs?
Red versus green, blue versus yellow, and light versus dark. Think of it like a seesaw. A single neuron, or a small group of them, handles the red-green balance. If it tilts toward red, it cannot signal green at the same time.
Which is why reddish-green isn't a color we can imagine. It's a biological impossibility. It's like trying to say north-south.
Exactly. And this opponent mechanism is responsible for that cool party trick, the afterimage effect.
Oh, I've done this. You stare at a green dot and see a magenta one after.
Yep. If you stare at a red square for 30 seconds, you are just hammering the red side of that seesaw. You're fatiguing those neurons, depleting the chemicals needed to send that signal. Then, when you look at a white wall,
The seesaw snaps back.
it snaps back hard. The red side is exhausted and stops firing. So the green side, which is normally inhibited by the red signal, suddenly has free rein. It fires like crazy.
And you see a ghostly green square that isn't actually there.
It's your brain trying to rebalance its own internal scales, a biological reboot.
It's wild that we can so easily hack the system just by staring at something. But let's zoom out from the mechanics to the history, because we didn't always have this fancy three-color system.
We did not.
You mentioned earlier that mammals are generally pretty bad at color compared to other animals.
We are, for the most part. The standard mammalian condition is dichromacy 2 cones.
Like dogs and cats.
Exactly. Dogs, cats, cows, horses, they basically see a world of blues, yellows, and grays. They don't distinguish between red and green. To a dog, a red ball on a green lawn is just 2 different shades of gray.
Why? I always thought evolution was about constant improvement. Why would mammals lose colors?
Because of the dinosaurs.
The dinosaurs.
For over 100 million years, mammals were small, nocturnal creatures. We were living in the shadows of giant reptiles, trying not to get eaten.
So we went through a massive nocturnal bottleneck.
Precisely. And in the dark, color is useless. What you need is light sensitivity. So early mammals ditched 2 of their 4 ancestral cone types to make room for more rods. We traded color for survival in the dark.
So we basically spent the entire Jurassic period seeing in black and white, just hiding in holes.
More or less. But then, much later, about 30 or 40 million years ago, a specific lineage of old world primates, our ancestors, did something remarkable.
What was that?
They re-evolved a 3rd cone. A gene duplication event occurred, and we split that single yellow sensitivity channel into 2 distinct pathways, one for red and one for green.
And the big question in evolutionary biology has always been, why? What was the pressure that forced us to get that specific color axis back?
Exactly. What was so important about red and green?
Well, the story I always heard was fruit. A monkey needs to see the bright red berry against the green leaves. If you can't see the fruit, you starve.
That's the classic fruit theory, and it holds up very well. Fruits co-evolved to be conspicuous, to be easily seen by the one animal that would eat them and carry their seeds somewhere else.
A perfect symbiotic relationship.
But there is a competing or perhaps a complementary theory that's gained a lot of ground. It's been pushed by researchers like Dominy and Lucas.
Okay.
They suggest it wasn't just about dessert, it was about the main course. It was about the salad.
Salad. You mean leaves. All the leaves are green.
Not all of them. In many tropical plants, the new young leaves are flushed with red or orange pigments called anthocyanins.
I think I've seen that on houseplants.
Right. Those young reddish leaves are tender, easier to digest, and much higher in protein than the tough, mature green leaves, which are often full of toxins.
Ah, so when fruit was scarce.
Being able to spot those reddish, nutritious young leaves against a sea of green canopy was a matter of life and death.
So the ability to appreciate a Rothko painting might stem from a monkey needing to find the one edible leaf in a forest of toxic ones.
It's a survival tool. We treat color as an aesthetic pleasure, but biologically, it's data. It's a nutrition label written on the world.
This idea of color as a tool brings me to something I found really controversial in the reading. We talk about human vision as if it's one single thing. But there's studies suggesting that men and women might literally see the world differently, dating all the way back to these survival roles.
This brings us to the hunter-gatherer hypothesis. It's a fascinating and, yes, very debated area of evolutionary psychology and biology.
So what's the core idea?
The premise is that for vast stretches of human history, there was a general division of labor. Males were predominantly hunting, females were predominantly gathering.
I've definitely seen this argued about online, usually with people claiming it's just a social construct. But is there any evidence for actual hardware differences in our eyes?
There seems to be. The hypothesis suggests that the hunter role required specialization in tracking fast-moving objects, often at a distance. You need to see the gazelle breaking cover 200 yards away.
Motion detection.
Right. And this aligns with a specific neural pathway in the brain, the M cell pathway, which handles motion and depth, but is largely colorblind. And studies show men tend to perform slightly better on these kinds of motion tracking tasks.
Okay, so that's the motion part for the hunters. What about the gatherers?
The gatherer role requires the opposite skill. You need to spot stationary objects, berries, tubers, specific plants that are camouflaged in a complex static background.
That sounds harder.
It relies on the P-cell pathway, which is specialized for object identity and, crucially, for color. And the genetic evidence for this potential split is surprisingly strong.
This is the X chromosome thing, right? I know color blindness is a guy thing,
Yeah.
but I never really understood the genetics of why.
It's very simple genetics, actually. The genes that code for the red and green photopigments are located on the X chromosome.
Okay.
Men are XY. They only have one X chromosome. If that one X has a typo of mutation that messes up the red or green pigment gene, they are colorblind. Game over. There's no backup copy.
But women are XX.
Exactly.
Yeah.
So if a woman inherits one faulty X, she has a backup X with a healthy gene, she won't be colorblind. But here is where it gets really, really cool. And this links directly to that Murray et al. Study from 2012.
I'm listening.
Because women have two X chromosomes, they might carry two slightly different but perfectly normal versions of the red or green gene. One might perceive a green that is slightly more lime, and the other a green that is slightly more forest green.
So they have a broader, more nuanced palette baked into their genetics.
Potentially. And Murray's study looked at peripheral vision. They tested how well people see color out of the corner of their eye.
And what did they find?
In men, as you move a colored object to the side of their head, their perception of color saturation drops off fast. A green object starts to look whitish or yellowish.
But not for women.
The study found that females maintained color sensitivity much further into the periphery. Their color world is whiter.
This sounds like the fridge blindness phenomenon. My wife can spot the ketchup bottle on the door shelf without even turning her head, and I'm standing there staring straight at it, swearing it's not there.
It might literally be that your peripheral M cones are failing to give you the saturation data that she's getting. Her visual field is painted with more stable color than yours is.
And there's an even more extreme version of this, isn't there? This idea of tetrachromacy.
Yes. This is the superpower that could emerge from that XX chromosome advantage. Since the red and green genes are on the X, a small percentage of women might inherit a mutated gene for color blindness on one X and normal genes on the other.
So they aren't colorblind.
They aren't. But what might happen is that they express both versions of a pigment gene. Instead of the standard 3 cone types, they might actually have 4 functional cone types.
4 functional cones. What would that even look like?
We can barely imagine it. A true functional tetrachromat, someone whose brain has learned to use that 4th channel of data, could theoretically distinguish up to 100 million colors.
100 million. Compared to what for the rest of us?
About 1 to 10 million, depending on the person. They might look at a beige wall and see a subtle ripple of 10 different distinct shades where you and I just see beige.
It's like they have access to a secret layer of reality.
It really is.
Speaking of layers of reality, I want to pivot to the layer we've now built for ourselves. We evolved for the savanna green leaves, red fruit, full spectrum sunlight, but now we live in boxes lit by LEDs and stare at screens for 8, 10, 12 hours a day.
This is the great modern mismatch.
Yeah.
We have taken a visual system that was exquisitely tuned for the sun and placed it in a completely alien environment.
I want to picture a specific scenario. Take a guy, let's call him Elias. It's 2 a.m. He's a programmer. He's bathed in that sterile, bluish glow of his monitor. He's not sleepy, but his eyes hurt, and he feels this weird, vibrating kind of wired but tired feeling.
A very, very common scenario.
Right. And reading the research, it seems like Elias is suffering from a profound confusion of his most ancient biological sensors. What is actually happening to him?
To understand Elias's headache, we have to talk about a third type of photoreceptor in the eye that we didn't even know existed until about 20 years ago.
Not rods or cones?
No. They are called IPRGCs, intrinsically photosensitive retinal ganglion cells.
That is a mouthful. What do?
They don't help you see in the sense of forming an image. You can't read with them. You can't see color with them. They're purely light intensity meters for the body's clock.
A light meter from my brain.
Exactly. They contain a special pigment called melanopsin, and they are tuned specifically to detect blue light right around the 460 to 480 nanometer range.
So they are, for all intents and purposes, blue light detectors.
Yes, and their only job is to tell a part of your brain called the hypothalamus what time of day it is. When high-intensity blue light hits these cells, they send a powerful signal saying, "It is high noon, the sun is directly overhead, be alert, suppress melatonin."
So when Elias is staring at his computer screen at 2 a.m.,
he is blasting these ancient cells with the exact frequency that screams daytime. His conscious brain knows it's the middle of the night, but his IPRGCs are telling his endocrine system it's noon. This creates a massive physiological conflict.
But it's not just the timing of the light, is it? It's the quality. The sources talk about the sun being a black body radiator. What does that mean?
It means the sun emits a smooth, continuous curve of all colors. There are no gaps. It's a full, balanced meal of light. Modern LEDs, on the other hand, especially the cheap ones in offices or on our screens, are spiky. They have a very weird spectral output. They give you a massive spike of blue energy, then a dip in the cyan, a spike in the yellow and green, and almost no deep red at all.
So it looks white to our eyes.
It looks white, but it's actually junk food light. It's spectrally empty. It's like trying to live on a diet of only sugar and salt.
And that's what's causing the eye strain for Elias.
It's a huge part of it. We are subjecting our primitive biology to a light diet it never evolved to handle. That sense of visual fatigue or unsettledness he feels is likely his visual system struggling to focus and process this unnatural, spiky spectrum. We are effectively giving ourselves jet lag every single night without ever leaving the room.
It makes you want to throw out all the smart bulbs and just go back to candles. Candles are black body radiators, aren't they?
They are. Firelight is the original light we co-evolved with after the sun went down. It's very warm. It has almost no blue light in it, so it doesn't trigger that wake-up signal from the IPRGCs.
So we've covered our internal hardware, our deep evolutionary history, the differences between us, and how our own technology is now hacking our biology. But I want to end by looking outside our species entirely.
Good idea.
Because as cool as our vision is, we're kind of in the minor leagues of the animal kingdom.
We really are. We think we're the pinnacle of creation, but compared to birds or reptiles, we're way behind. Many birds are tetrachromats. They have that fourth cone we were talking about, but theirs is for ultraviolet light. UV.
So a bird looks at a forest and sees things that are literally invisible to me.
It sees a completely different map of reality. Flowers that look plain to you have these intricate UV bullseyes on them, guiding bees and birds directly to the nectar. But birds also have another incredible adaptation called oil droplets.
Oil droplets, like in their eyes.
Inside their actual cone cells. Each cone has a tiny microscopic drop of colored oil that sits in front of the photoreceptor, acting like a filter.
Like putting a yellow lens on a camera.
Exactly like that. It's like a pair of high-performance sniper goggles for each cone. It cuts out all the fuzzy, overlapping wavelengths and sharpens the signal. This allows birds to distinguish colors with a precision we can't even dream of.
So where I just see green, a bird might see 20 distinct shades of emerald, olive, and chartreuse, all perfectly separated and defined.
Precisely. And they use this for mating. To us, a male and female blue tit bird might look identical. But to them, the male is glowing with UV ornamentation on his chest that screams, "I'm a good mate." We are blind to the secret codes of nature all around us.
And then there is the mantis shrimp, the internet's favorite crustacean. The notes say they have between 12 and 16 types of photoreceptors. 16, we have 3. Why do they need 16?
This was a huge mystery for a long time. Scientists thought, wow, they must see a rainbow of millions and millions of colors we can't even fathom.
That's what I would assume.
But the recent research suggests they don't actually process color the way we do at all. They don't do the algebra of comparing the outputs of their cones.
They don't mix the paint.
No. With 16 distinct narrow sensors, they don't need to mix. They just register the raw input. It's like a barcode scanner.
A barcode scanner?
Yes. They scan the coral reef, and the specific pattern of 16 inputs tells them instantly edible crab or poisonous rock or potential mate. It's not about aesthetic beauty. It's about raw processing speed. They are seeing the world in a way so alien that we can't really comprehend it.
It brings us right back to that tomato in the produce aisle at the very beginning.
It does.
The dog sees a gray shape. The bird sees a UV glowing beacon. The mantis shrimp sees a specific 16-digit barcode. And we see a red fruit.
And none of us are seeing the truth. That's the absolute key. Evolution doesn't care about truth.
It cares about fitness.
It only cares about fitness. If seeing the truth, the chaotic quantum soup of energy, helped us survive, we would see it, but we don't. We see red tomato because the label red tomato tells us, calories here, eat this, survive.
Color is just a user interface. It's the desktop icon for food.
That's the perfect analogy. You don't need to know how the electrons in your computer's processor are moving to write an email. You just need to click the blue envelope icon. Our entire reality is just a desktop of icons that lets us survive the day.
That is both incredibly humbling and profoundly mind-expanding. We are operating a biological machine that sketches, paints, and color corrects the universe for us in real time.
And doing a pretty good job of it for the most part, despite the glitches.
Before we go, I want to leave everyone with one thought that's been nagging at me since we talked about the birds and the shrimp. We know that color is just a translation of data, and right now we're limited to this tiny sliver of the electromagnetic spectrum. But we build machines, sensors, that can see X-rays, infrared, radio waves, Wi-Fi signals. We have the hardware. The thought is, what happens when we figure out the wetware? What happens when we interface those sensors directly into the visual cortex? If we could suddenly expand that dark theater in our minds to include the entire electromagnetic spectrum, we might stop seeing objects altogether. If you could actually see the Wi-Fi signals passing through the walls and the radio waves bouncing off the atmosphere, the whole illusion of a solid, stable reality could simply dissolve. We might finally stop seeing the icons and start seeing the underlying code. And if we did that, our very definition of what it means to be human would have to change entirely.
That is the ultimate question. The future of vision might not be about seeing more clearly, but about seeing something else entirely.
Thanks for taking this dive with us. We'll see you next time.